Right, so hi everyone. Thank you for joining our BrainMap webinar today. I'm very pleased to introduce today's speaker, Professor Mara Cercignani. Mara is the academic director of the Clinical Imaging Sciences Center at Brighton Sussex Medical School, where she also leads the MRI physics group. She studied telecommunication engineering at the Polytechnic of Milan and completed her master in MRI at Leicester University. She then returned to Italy, where she works in Milan, before moving to London, where she completed her PhD in quantitative MRI at UCL. Her research focuses on quantitative MRI, including in particular diffusion MRI and magnetization transfer MRI. Mara is interested in all aspects of quantitative MRI, from modeling of signal to sequences design and image processing, with a specific aim of translating physics development into clinical applications. Today, she will talk about advanced MRI for in vivo imaging of the effects of inflammation on the CNS. So before I hand the mic over to Mara, I just wanted to remind that please address your questions using the Q&A box and not the chat. If you want to ask the question yourself, just put your name and we'll unmute you. Otherwise, we can just read the question out loud, either during or at the end of the presentation. Right, so without further ado, we'll turn it over to Mara.
Thank you very much. Thank you for the invitation. It's really a pleasure. I guess it's one of the good sides of this situation is that the interactions between labs can be a little bit more long range than normal. So I'm going to be talking about some of the research that we have done here at Brighton, which is a town south of London in the UK on the coast of the English Channel, for those of you who don't know. Yes, I'm going to be talking about imaging of the effects of inflammation in the central nervous system. So the motivation really for doing this work is that inflammation and more specifically neuroinflammation is obviously something that happens in a lot of conditions that affect the central nervous system. And even going beyond the diseases that are classified as inflammatory, such as multiple sclerosis, we know now that it has a prominent role also in neurodegenerative disorders and in some psychiatric conditions. And particularly, it's been implicated in major depression. It's not easy to study in humans because we don't have any obvious biomarker for inflammation, and animal models are not always suitable. And the other thing that we need to consider is that when we talk about neuroinflammation, really we have, this is a really broad term that encompasses a lot of responses from the CNS to injury and infection. So some people, when they talk about neuroinflammation, might be thinking of something similar to what represented in this cartoon, where you have a leakage of the blood-brain barrier and therefore infiltration of immune cells into the brain, including monocytes and T cells, and then consequent activation of resident microglial cells, which might cause, for example, damage to the myelin, such as happens in multiple sclerosis. But there is also a concept of neuroinflammation that is much more subtle when we don't have this direct influx of immune cells in the brain, but we have some activation of the resident microglial cells anyway. And the particular situation that has gained a lot of interest in the past few years is something known as a sickness behavior. So this sickness behavior is essentially a combination of behavioral symptoms that people experience when they have inflammation somewhere in the body. So this is something similar to what every one of us has certainly experienced when they have the flu. So a combination of symptoms that can be vary in seriousness that include fatigue, anhedonia, loss of concentration, apathy, and because of the characteristics of these symptoms, it is reasonable to assume that there is some mediation of the central nervous system. And this seems to be an evolutionary type of behavior that has developed to prevent people from going out and socialize when they are sick, essentially a way of preventing communication of disease when people are experiencing infection from an external pathogen. So in a way, we could see this as a natural mechanism to impose self-isolation on people. What is interesting about this is that in this case, the inflammation is not in the brain, the inflammation is in the body, but there has to be a way that this is communicated to the brain because otherwise people wouldn't be experiencing this kind of symptoms. So this is the sort of inflammation that has mainly been implicated in conditions such as major depression, and we will talk a little bit more about this later. So how to image the effects of inflammation on the brain? Well, what we know about this condition of inflammation is that certainly there is some activation of certain glial cells, particularly the microglia. And one way of measuring activation of microglia is through TSPO PET. So TSPO is the translocator protein, which lives in mitochondrial membranes and tends to be overexpressed in activated microglia. So it is possible to image activated microglia using TSPO PET. And this picture that you see on this slide shows what happens if you take a TSPO PET of people at baseline and then you inject them with LPS, which is an endotoxin, which is recognized by our immune system because it typically wraps around bacteria. So it triggers a fairly strong immune response. And you can see that if you repeat the PET after injecting people with LPS, you can see that there is a much stronger uptake of TSPO ligand basically everywhere in the brain. So this method works, but is difficult to perform, and it tends to be expensive, and it requires injecting people with radioactive tracers. So we are interested in developing MRI methods that can detect inflammation. Obviously, it cannot be such a direct way of imaging inflammation. It has to be somehow more indirect. So we started off by looking at what the typical features of inflammation are in the brain. So you can have some of or all of these things happening during your inflammation. You can have an increased blood-brain barrier permeability, although this only occurs really when the focus of inflammation is in the brain. You can have an increase in glial density and activation, typically of microglia and astrocytes. You can have an increase in the concentration of pro-inflammatory cytokine. And also you can have a change in the water volume fraction, and in particular an increase in the extracellular volume fraction. And then you can have a series of other things happening, typically more downstream, that form the post-injury repair mechanism. So the question is, can we measure any of these using MRI? So obviously we can do something about some of these, and in particular, obviously we can use contrast enhanced MRI to measure blood-brain barrier permeability using gadolinium, and even the infiltration of macrophages using iron oxide particles. But we can also try to use non-contrast MRI to look at things like changes in the size of the extracellular water compartment. And this can be done, for example, using magnetization transfer MRI. And I will show you that maybe using a quantitative version of magnetization transfer, you can also start to look at effects that are probably linked to metabolism. And you can also think of using, obviously, diffusion, MRI, and MRS to look at changes in cellularity and potentially in glial morphology. So I'm going to talk specifically about these techniques because this is what we've been concentrating on in the past few years. And I'm going to show you a few studies where we've tried to look at the effects of inflammation using these techniques. So I know that most of you are imaging experts, so I'm just going to give a very brief explanation of what magnetization transfer is. is essentially an interaction that occurs between hydrogen protons in the water and the hydrogen protons that are attached to larger macromolecules. So the latter protons are a lot less mobile, but they can come in contact with the water and interact with them and affecting their magnetization status. So typically, for the purposes of modeling the signal, we assume that there are two pools of protons, one which is the liquid pool or the water pool, and this is the pool of protons that normally produces the MRI signal, is fast moving and has a relatively long T2 compared to the typical echo times of an MR experiment, and the narrow line in spectrum. By contrast, the semi-solid or macromolecular pool is slow moving and has very short T2. This corresponds to a wide line in spectrum. So if we want to represent very schematically the spectra of these two pools, they would look something like those shown in this picture, where you have the narrow spectrum of the free water and the wide spectrum of the macromolecular pool. And to do an MT experiment, what we want to do is somehow act on the macromolecular proton without acting directly on the free water proton. And this can be done using radio frequency pulses that are tuned at a frequency far from the Larmor frequency. In this way, we can saturate the macromolecular protons without affecting directly the free protons, but because of the exchange of magnetization that exists between these two, this will reflect in a partial saturation of the free water, which will cause a signal attenuation. So this signal attenuation will be somehow related to the density of macromolecules in a given tissue, and also to the efficiency of exchange of magnetization that exists between the two pools. So this mechanism of contrast has been used for many years, also to obtain semi-quantitative maps of the so-called magnetization transfer ratio, or MTR, which is simply calculated by computing the percentage difference in signal between an image acquired with MT saturation and one without. So an MTR maps will look similar to this picture shown here in the center of the slide. And its main characteristics is to have a little bit of contrast between white matter and gray matter. Typically, highest values are found in structures such as the corpus callosum. And in the gray matter, the highest values are typically found in the thalamus. This is all consistent with a larger proportion of proteins in the white matter, which is justified by the fact that the white matter is rich in myelin. And so myelin is considered the main contributor to the MTR signal in the white matter. Because of this, obviously, MT has been used a lot in the study of multiple sclerosis, even in the late 90s. And but it's important to remember that even though the MTR has been associated with myelin content, it always measures it as a fraction of the total water. So, being it fundamentally a ratio, its value can change if the numerator changes, but also if the denominator changes. And therefore, if we think that inflammation might result in an increase in extracellular volume fraction, then we will observe a decrease in MTR. And this can be perhaps understood by looking at this quite old paper that looked at the evolution of MS lesions, multiple sclerosis lesions over time. So in this study, patients were scanned regularly every week, and the MTR was calculated. And when a new lesion appeared on gadolinium-enhanced scans, researchers would just go back to up to a month before the lesion appeared and measure the MTR in the location where the lesion would later appear. And so this is how they built these patterns, and they identified 4 different patterns of MTR variation in MT lesions. But rather than being interested in these patterns, what I want to point out is that for all the patterns, there is a time, approximately a week before the appearance of the lesion, where you see a dip in the MTR, and that's precisely an indication of the inflammation that is taking place. And here we're talking about frank neuroinflammation. on a more quantitative side, Greg Stanich did a nice experiment looking precisely at the sensitivity of magnetization transfer to inflammation, which consisted of injecting TNF-alpha in the sciatic nerve of rats, and this triggers a marked inflammatory response with very minimal effect on the myelin and the nerve itself. So it's a good model for studying pure inflammation. The MRI was performed after 2 days and then compared to histomorphometry and immunohistology. And what they observed was that there was a really nice linear correlation between the MTR and the extracellular volume fraction, and that these two divided quite nicely the treated nerves with TNF-alpha and the untreated ones. So these data together supported the idea that MTR is sensitive to inflammation. However, it is also sensitive to a lot of other things. And the MTR itself is a very crude measurement. So if you were to perform lots and lots of magnetization transfer experiments, changing the way that you saturate your macromolecular pool, so changing the power and the offset frequency of the saturating pulses, you will end up with curves that look like these ones. This is known as a Z spectrum. And basically every point represents the signal measured at a different saturating power and different offset frequency. So the powers are shown by the different colors, and the horizontal axis shows the offset frequency. And when we calculate the MTR, what we're doing really is simply taking two measurements in this plane and calculating the difference or percentage difference between them. But looking at this Z spectrum in its entirety, we see that by doing that, we're really throwing away a lot of information, and perhaps by trying to characterize the signal produced by MT saturation a little bit more, we can certainly extract more information. So that's what prompted really the development of quantitative magnetization transfer, where instead of just looking at the ratio between the images, we're trying to model the signal and through the parameters that might affect it. So still starting from the concept of the two pools, the macromolecular and the liquid pool, in this case, what we try to do is we characterize each pool by their relaxation times, T1 and T2, and by the rate at which they are able to absorb radio frequency energy from the saturating pulse. So those would be RRFB and RRFA, and also by their forward and exchange rates, which in this cartoon are represented by KF and KR. So if we then write the coupled block equations for this system, it is possible in some special circumstances to actually find an analytical solution for these coupled block equations, and therefore we end up having a signal equation that we can fit to our signal, providing that we acquire sufficient data points varying the acquisition parameters. And if we do that, we end up building quantitative maps of some of the MT model parameters. And in this picture here, you can see some maps of the forward exchange rate, KF, of the pool size ratio, F, which is essentially the ratio of the spin density in the macromolecular pool and the spin density in the liquid pool. So this F parameter is the one that has gained more interest in the field of magnetization transfer because it's been often considered a proxy for myelination. T2F is essentially the transverse relaxation time of the liquid pool, which is very similar to the T2 that you would measure using other quantitative T2 mapping methods. So contrary to the old myelin field, in this context, we're more interested in KF and possibly T2F for our purpose of measuring the effects of inflammation. So we have hypothesized that because of, well, T2F sensitivity to water increase and KF sensitivity to metabolic properties, they might somehow be sensitive to the effects of inflammation. And this was tested in this study, which we did a few years ago, involving 20 healthy people. So they came in for a baseline scan, and then they received an injection. So the injection could be either a typhoid vaccination or a placebo. So that would be just saline. Typhoid vaccination is the same vaccination that you would get if you were to go to a country where there is a risk of contracting typhoid. And it triggers a very mild immune response, so like all vaccinations. So this was a way of modeling peripheral inflammation and see its effects on the brain. So after 3 hours, people would receive their MRI scan, and then they would return after about a week, repeating exactly the same procedure, except that at this time they would receive the injection that they didn't receive the first time. We also collected behavioral data and blood samples at baseline and at 3 hours. And obviously the MRI session included the QMT scan. In parallel, we also ran an FDG PET study. So FDG PET measures glucose uptake. I have to stress that the participants in the two studies were not the same. So we can only compare the results in broad terms, but not very specifically. But the idea was to measure the same effects. So in this case, people came for their scan. They had a PET scan and the usual assessments, and then they received the injection, which could be typhoid or placebo. After 4 hours, they received a second scan, and then a second injection, which was the other one. And then finally, at 8 hours, they received another scan. So slightly different design, but essentially looking at the same thing. And these data here show you that even though the reaction to the vaccine is mild, it does produce an increase in IL-6 in the blood. So you can see the red bars show the results for the vaccine session, while the blue ones are the placebo session. And you can see that there was absolutely no change in this pro-inflammatory cytokine in the blood when receiving the saline injection, while there was a significant change when receiving the vaccine. And also in terms of feeling of the participants, you can see that there was a significant increase in perceived fatigue in participants when they received the vaccine, and a lot less when they, although we still see an effect, but it's non-significant when they received the placebo injection. So what was interesting about this was that we actually found an increase in one of the MT parameters in the insula. And this area of the increase of, and the parameter that was increased was KF, so the forward exchange rate. And what we also observed that this change in KF was mildly correlated with the change in reported fatigue, self-reported fatigue by the participant. And this is very interesting because the insula is the main, one of the main regions in the brain that is involved in the interoceptive pathways. So these are the pathways that regulate the communication between the body and the brain. So along these pathways travel the signal that tell the brain about the body status. So clearly when you are sick, it's very important that your body communicates to your brain that you're sick, and it looks like there might be something happening in that particular area of the brain when there is some inflammation happening in the body. And the role of the insula in this, in receiving communication about bodily status is very well known and very well documented. Here are reported some of the studies that have looked at things like you know, pain, exercise, itch, cold, things like that, all this kind of sensations that happen in the body. And when they happen, the insula is the region that either activates or somehow changes in response to this change in the bodily state. And interestingly, now here on the left-hand side, you can see the MT results that I've just shown you. And on the right-hand side, you see the result of the FDG PET study. And these are the areas where there was an increase in glucose uptake. So an increase in metabolism in the PET study. So you can see that the regions are very similar anatomically. As I said earlier, these were not the same participants, and therefore we can't do a subject by subject comparison. And obviously the PET results seem to be bilateral, while in the MT we only had the left hemisphere, but of course this might just be down to statistical power issues. But in general, it is quite striking that we do completely different methodology that we think both measure somehow changes in metabolism, we see an effect in the same anatomical region. And one of the possible interpretations for these changes in KF is that there is an increase in the anaerobic glycolysis, which leads to an increase in lactate, which obviously contains some OH groups and therefore might be somehow detected by MT technique. So this study led us to believe that perhaps we were onto something with these MT studies, and we wanted to look at a different model of inflammation. And so we turned to interferon alpha. So interferon alpha, until very recently, was the treatment of choice for hepatitis C. And it's very effective in treating hepatitis C. The problem is that in about 40% of people treated with interferon, they start to manifest the symptoms that are undistinguishable from those of major depression. And this is the most substantial evidence towards the current theories of inflammation in major depression. So in this study, we recruited people who were due to start their treatment with interferon alpha. So on the day that they were due to start their treatment, they would come to our imaging center and receive an MRI scan and the usual neuropsychiatric assessment. And then they would start their treatment and receive another scan after 4 hours from the beginning of the treatment. And then we just did the normal follow-up, so without MRI, just the various assessments after 4 weeks, 8 weeks, and 12 weeks. So here you can see in this graph that most of our participants developed depression after the treatment. It wasn't measurable after 4 hours, but it was definitely measurable after 4 weeks. And this is consistent with the data shown by others. And what we found from our imaging data was that there was, again, an increase in KF, the same parameters that we found increased in the insula in the previous study, but this time the changes were mainly located in the basal ganglia area. We also found a decrease in T2F. And this was measurable when comparing the whole brain, and obviously even more when doing just the region of interest focused on the basal ganglia. And in that case, it became bilateral. So, excuse me, one of the reasons for focusing on the basal ganglia was that others had shown before that the effect of interferon alpha might be measurable in those areas. So these two studies were done, one with MR spectroscopy, looking at changes in glutamate, and the one at the bottom is a study with FDG PET, and they both showed that the basal ganglia, in particular the putamen, might be sensitive to the effects of inflammation. And this is consistent with some hypotheses about how how interferon reaches the brain through some animal studies. And you might be wondering why in the typhoid vaccination we found changes in the insula, while in this case we found changes in the basal ganglia. It's not completely random, because it is known that there is more than one pathways that allow communication between the body and the brain. And it is possible that it might depend on the strength of the immune system response, or in the type of infection. So in this case, we are looking at a more viral-like versus bacterial-like infection. And in our studies, we've always found that the first one, so we typically observe that one tends to be involved primarily the interoceptive pathways and therefore the insula, while the other tends to involve more the basal ganglia. Excuse me, I'm going to move on to the studies that we've done looking at the utility of diffusion to measure inflammation. I'm sure that all of you are very familiar with the diffusion MRI, so I'm not going to spend the time explaining it. Of course, measures the random motion of water molecules within tissue, and because this random motion is affected by the obstacles encountered by these molecules, by measuring diffusion in the brain, we can infer indirectly about the microstructure of the brain itself. So because of this, we can also start to think about how within a single voxel, there might be more than one environment, more than one compartment. And if they are characterized by different diffusion properties, then through modeling, we're going to be able to distinguish between them. So this is interesting from the point of view of measuring inflammation, because if we imagine that there is a change in the size of these compartments, or a change in the cellularity, or other effects like those, then we should definitely be able to pick them up using diffusion MRI. So we have tried to use NODI, neurite orientation, dispersion and density imaging, to look at this, because it's one of the simplest methods available out there to look at diffusion compartmentalization. And we are hoping that whatever methods we look into, they are usable in clinical populations. So we wanted to use approaches that are immediately translatable to the clinic. And NODI is, because it's so simple to do and the acquisition is not extremely demanding. So the idea of NODI is that the signal in each voxel can be split into 3 compartments: the isotropic diffusion, which is mainly in the CSF, intraneurite water, which tends to have restricted diffusion, and the extraneurite, which is modeled as hindered diffusion. So we have looked again at the interferon model. So this was a continuation of the same study, but only 7 of the people included in the NODI part of the study overlap with the ones included in the MT study. But the design, of course, was exactly the same as described before. And among the assessments that we took from these people, there was the visual analog scale of fatigue. So essentially, this consists of asking people at different times, how fatigued do you feel between 0 and 100? And they basically just put a line at the point where they think they are. And based on our previous studies, we had the 2 main regions of interest, which were the insula and the striatum. And it was quite disappointing because we found no main effect. So when comparing the after interferon diffusion data with the pre-interferon diffusion data, there was no significant effect. So there was a little bit of a consolation, because when we looked at the correlation in the intraneurite volume fraction and the change in fatigue from the self-reported VAS, we found that there was a significant association. However, this obviously is a fairly small result without having that main effect. So I reported here the results from the MT study, just to show you that they very nicely co-localize. So we started wondering why that might be, why we didn't see anything. And one of the things that we've been thinking about is that obviously diffusion MRI is sensitive to the features of the intracellular space, but it cannot distinguish between types of cells. Water is in all the cells, is in neurons, is in glial cells, and so on. And the signal in diffusion is likely to be nominated by the most abundant or the larger of these cells. So since the most interesting cells in the context of inflammation are microglia, which are very small, and there aren't that many of them, it's difficult to expect to observe results unless you do some very sophisticated modeling of the signal, which people are doing, by the way. So we turned to diffusion-weighted MR spectroscopy, where the idea is that we're still looking at diffusion, but a diffusion attenuation that we measure on the metabolite peaks. So thanks to the fact that the main metabolites can be somehow found primarily in certain types of cell, we can start to distinguish between changes in the intracellular space of glial cells and neurons. So for example, we know that NAA is exclusively in neural cells, while choline is in both glial cells and neurons, but most abundantly in glial cells. So there's more choline in glial cells than in neurons. Creatine is in both. So we did a very small study that unfortunately is still ongoing, but we had to stop recruitment for obvious reasons. In this case, we wanted to use a much more prominent inflammatory challenge. So we used the LPS, which, as I explained before, is an endotoxin that triggers a very, very strong immune reaction. It only lasts for a few hours, but is very noticeable. And the idea here was to look at the effects of LPS injected peripherally on diffusion-weighted spectroscopy. The hypothesis was that if LPS has an effect on microglial but not on neurons, then we should observe changes in the ADC of choline but not that of NAA. So the whole study so far has recruited 16 participants, but unfortunately only 7 of those had diffusion-weighted MRS. And they spent 2 days with us, about 2 or 3 weeks apart. During each session, they received an injection, which could be either LPS or placebo, randomized. And they received a lot of assessments throughout the day, and they received a TSPO PET and MRI that included QMT and diffusion spectroscopy. So these data here are just to convince you that this really is not a walk in the park, this LPS injection. If you look at the bottom row, these graphs show physiological changes happening. So in red, the red curve shows the data from the LPS session. This is averaged across all the 16 participants. So you can see that there is a sharp increase in body temperature already about before 2 hours after the injection. And the people do feel sick. They really are, like they have a really bad flu. And there's an increase in the BPM and also a decrease in heart rate variability. And from a subjective point of view, you can see that they become more depressed. The POMS is a scale that measures mood. There is an increase in fatigue and an increase in the sickness scale as well. So this is to convince you that this really is a good model of peripheral inflammation. Some more information about the diffusion spectroscopy. We looked at two separate voxels, which were, one was positioned in the thalamus, and this was our main region of interest, which was chosen because it's an area rich in TSPO receptors, and the long-term plan is to compare this data with the TSPO PET data. And the other region was placed in the white matter around the corona radiata. And interestingly, even with such a small number of participants, we found a very significant increase in the ADC of choline in the thalamus, while everything else stayed the same. We didn't observe any other change. There was a very small increase in the ADC of creatine, but this was not significant, and it was expected because creatine is present in both glial cells and the neurons. There was absolutely no change in the NAA. I should also point out that we looked at the peak area in the B0 spectra, and there was no change there. So the change in the ADC is really a change in the diffusion coefficient, is not an indirect effect of a change in the density. So this is interesting because the hypothesis there was that when microglia become more activated, they change their shape. So it is possible that through this we're measuring a change in morphology. Of course, it's very speculative for now because this parameter is not, so choline is not only microglial, it's in all glial cells, and obviously astrocytes are much larger and many more than microglia, so we could be measuring astrocytosis. So the jury is still out, and we need obviously more data to substantiate this, but it's such a clean result that I was keen to show it. We also found an association with self-reported mood, both the total mood score and the negative mood. Obviously, 7 participants is a very small number, and obviously there are some outliers, so these results should be taken with a pinch of salt, but very promising so far. So I've basically come to my conclusions, which are going to be that, so we can't really measure inflammation in the brain directly, or the effects of inflammation in the brain directly with MRI, because in the end MRI measures the signal from water, and therefore it's really difficult to make it somehow specific to the effects of inflammation. But what we can do is measuring events that typically accompany inflammation. And we have done that with 3 different techniques. We have found some results that altogether seem to build the story. Of course, these measures are very indirect. From the data that I have so far, I would say that the diffusion weighted spectroscopy really looks very promising, but of course it's got other limitations, and in particular the fact that so far at 3 Tesla we are bound to very large voxels, which might not be ideal. And the interpretation of the results is not always as straightforward because we're looking at something that is so indirect. And obviously, one of the biggest issue that I have sort of avoided so far is that when we look at models of inflammation like the ones that we've been studying, it's kind of easy to isolate the effects of inflammation and not worry about anything else, because we are just sort of probing the immune system and looking at what this does to the brain. But obviously in real diseases, typically there would be lots of things happening at the same time. So it's very difficult that in any disease to have just purely inflammation happening. And it's not clear at that point whether any of these techniques would be specific to inflammation or would just be affected by so many different pathological mechanisms that it becomes impossible to extract the relevant information. So that basically just goes back to the old point, which is that we really need to understand better what we are measuring when we use all these different MRI techniques, because they tend to be affected by many factors at the same time. So if you wanted to start to be really, really specific about inflammation as well as about anything else, we really need to go back and rethink some of our validation studies that are always invoked at many meetings on quantitative MRI and so on. And I can just join the choir in this. I want to finish by thanking my colleagues who have been really instrumental to all this work, in particular Neil Harrison, who used to be at Brighton and now has moved to Kubrick in Cardiff, and Alessandro Colasanti, who are really the promoters of all this research into inflammation. And also Itamar and Francesca, who have really helped with setting up the diffusion-weighted spectroscopy at our center, and all the others on this slide, who really did most of the work. I'm just summarizing the findings. So thank you. That concludes my talk.